Hello and welcome to a special edition TB Ed Talk for Hill Day Training. My name is Tori. I will be your host tonight, and this is TB Innovations and Breakthroughs with a bunch of special guests, including IAVI, TB Alliance, and TAG. We are actually just going to jump right in tonight. So, as always, if you've got questions at any point in these presentations, drop them in the chat. They will get over to me, and our incredible guests will be answering them towards the end of the chat stream. I know how to speak English today, guys. We're doing great. With that, I'm actually just going to get us kicking right off into the content because we have a lot of really cool stuff to cover tonight. So, oh no, which window is it in? There we go. All right, Lizzie, you're on mute, just so you know.
Thank you for letting me know. I had completely forgotten. Hi, everyone. My name is Lizzie Lovinger. I am the U.S. and Global Health Policy Director at Treatment Action Group. Really excited to be sharing some updates with all of you tonight, and excited to see you all in just a few short weeks. So we're going to get started with just kind of a review of all of the really storms that have kind of hit TB R&D in the past year, and that are important for us to know about as we kind of move through our presentation tonight. And I'm sure many of these you've already heard about in the media, but just to go over them once more, we saw these really broad scale award terminations, grant cancellations, and changes to how U.S. government funded R&D really operated in partnership with scientists. And so that came down to specific universities having their awards terminated, funding bans applied to foreign subawards. My daughter's coming to join us
Extra
right
special
now.
guest stars.
This is my special guest star. Funding bans specifically on South Africa or on universities collaborating with South Africa or China. And that, we'll talk about this in a little bit, but that was uniquely harmful for TB R&D because of the really amazing combination of circumstances that are in South Africa that are necessary for TB research to take place, including great infrastructure for research, a population that does have a high rate of TB, and great existing partnerships with academic researchers there. And also good governance, which is really necessary for facilitating research. There were also grant cancellations just based on executive orders and policies that were coming out of the White House. Things like DEI, vaccines, mRNA, those were all kind of considered targets for grant cancellations by the administration. There was also a pause on any foreign subawards, and we recently got an update on what the policy for new foreign award applicants is going to be, and it's incredibly opaque. Yes, you are. And there's really a lot of uncertainty amongst scientists about how that's going to work. I'm sure you heard about indirect cost limits, where the really necessary costs of conducting research that are not purchasing the research components themselves or paying the researchers, but are still really necessary to have the research operate, were limited to a point that really made it impossible for a lot of institutions. These are just a few examples, but really it's just been a prohibitively uncertain and restrictive and scary atmosphere for researchers this past year. Next slide.
I want to leave flag anymore.
So this is just really zooming in on the impact that has happened just as a result of that policy on no foreign subawards going to South Africa or any projects that are in partnership with South Africa, and just the outsized impact that this has had specifically on TB R&D, and really just why that has been so devastating to the R&D landscape. Next slide. So I'll try and do this as quickly as possible. And I think some of you might have seen a lot of this content in an earlier presentation, but just to kind of go over how TB diagnostics work for these traditional tests, it relies on sputum, which is very difficult to produce, especially for children. It's not just spit. It's you really have to kind of produce it from your throat, and it's difficult to do. It's also a really long diagnostic process, and that can lead to treatment interruptions or lost follow-up. And it's not as accurate as modern tests. So these newer molecular diagnostics are really kind of the gold standard. And they often use other biosamples like blood or urine or an oral swab, and they are much quicker. They can sometimes happen at the point of care or near the point of care, which is really important because in a lot of places where we see the highest rates of TB and where it's most important to be doing that diagnosis, there's not necessarily a sort of centralized lab. And all that is kind of sort of the second stage in diagnosis for a lot of people, where initial screening tests are needed before you can even reach that kind of diagnostic point. Next slide.
Hello, Mama.
So I'll sort of expand on why these point of care and near point of care tests are so important. And really, a lot of it is just improving accessibility to diagnosis. In the sort of continuum of care of TB, diagnosis is where we see the largest drop in where people with TB kind of get lost in the care cascade. So we want to be making testing and diagnosis as accessible and as effective and as fast as possible. So what would really make that as basically as point of care ready as possible is if something were battery powered, you don't need to plug it in, you have easy to collect sample types, it's heat resistant and durable. You can wear it out in the field. It doesn't need to be in like a very specific temperature controlled environment. And ideally, these simpler tests should be offered at lower prices. The trick in this has often been a kind of trade-off between decentralization versus testing sensitivity and specificity. So what those mean are testing sensitivity is the ability of a test to miss any false negatives, and testing specificity is the ability of a test to miss false positives. And both of those are obviously problematic for a number of reasons, and you really want to have tests be as sensitive and as specific as they possibly can. But what that often means is you kind of need a diagnostic platform to be further from the point of care. So there is this kind of trade-off that you're inherently making with a lot of the diagnostics that we have available. But even with lower test accuracy, an increase in accessibility would still increase treatment rates dramatically. So some of that trade-off to a lot of folks, I think, is kind of worth it. And all of this is to say that we want these new diagnostics to be able to work in children, to be accessible to children, and also work for extrapulmonary TB, which obviously sputum can't do. Next slide. As we see increasing drug resistance, we also want to focus on drug susceptibility testing. So that's really the test that tells us whether or not the type of TB that a person has will respond to TB treatment drugs or not. With the sort of increasing challenge of drug resistance, the WHO has called for universal drug susceptibility testing for all people accessing TB testing and diagnosis. But there are not any currently available drug susceptibility tests that are available at the point of care or near the point of care. So it usually requires positive test results to be referred, and then that would require more centralized lab access, which again delays the treatment timeline, which increases the chances of loss to follow-up. And all of this is really concerning when you're trying to address emerging drug resistance, including emerging resistance to bedaquiline, which is one of our newest available TB treatments and really the backbone of a lot of TB treatment regimens these days. And with that, I think I'm turning it over to Stephanie.
Thanks so much, Lizzie. This is a good segue to drugs because drug susceptibility testing is really important for drugs. My name is Stephanie Seidel. I'm from the TB Alliance. I just wanted to introduce our organization really quickly and talk about the space in which new TB tools are being developed. TB Alliance is a not-for-profit organization that's working to discover new drugs, develop them through clinical trials, and make them available at the country level, as well as working with countries to make sure that they can be adopted and taken up in a good time frame from when they're approved. Next slide. Like I said, we are discovering, developing, and delivering drugs at TB Alliance, but we're a not-for-profit small organization, which means that we can't do anything without an enormous network of partners all over the world. And this includes research partners that are in a lot of the countries that Lizzie was talking about, where there is a high burden of TB and where there is a lot of capacity to do research on the ground. And those locations and those partners are critical for moving research forward. And it's, it's very important to continue supporting that capacity development so that the research network is strong and continues to be strong. Next slide. I have a bunch of slides. I'm going to try and go through them relatively quickly, but I don't want to go too fast. Just to take a step back, I will see some of you in DC in a couple weeks. I'm looking forward to that. A lot of what we talk about is needing funds for TB drugs, for TB services, and we talk about the burden of TB, and it really is something we're still struggling with, and that has not changed in a long time, and we're still in a position where it's the deadliest infectious disease killer. But we wanted to just give you a sense of what's happening in the research field, because there are exciting things on the horizon, and there is a lot of opportunity to develop new tools that can make treatment easier, diagnostics quicker, hopefully prevent TB with vaccines. And this is what all of us have been working on across, TB Alliance, IAVI, and at Johns Hopkins. You'll hear from our colleague Kelly Curran about some other trials that are happening. But what we're looking at right here is a pipeline of drugs. So when I say we discover drugs at TB Alliance, we discover chemical compounds that may have activity against tuberculosis and that would be good in a regimen to treat TB. On the left, you see really, really early stage compounds that are going through really early stage tests, probably not in, in vitro or in animal studies, moving to preclinical development where there's, enough data that there is a higher likelihood that there would be appropriate to be in a regimen. And then moving into phase 1, 2, 3 in post-market studies, which is human studies and in patients that have TB, typically. You'll notice that there's a lot on the left, not very much in the middle, a lot of activities on the right, but with the same products. And this is because drug development takes a lot of different products to find one or two that really are going to be not just effective, but are safe and effective at curing the disease that you're curing. So this is what TB Alliance has developed, and I'm going to focus on the things that I have in circles here. Starting from the right-hand side, things that are already on the market that I wanted to give you updates about are pediatric formulations of drug-sensitive or first-line treatments. Protominid, which is a drug that TB Alliance developed but is used in a regimen called BPAL, bedaquiline, protominid, and linezolid to treat all forms of drug-resistant TB globally. And then I'm going to talk about some new compounds that are in development now, TBAJ876, and some new technologies like long-acting injectables. Next slide. Although we do have a really large pipeline of drugs, we are not the only entity that's developing new products. So this is what we call the global new TB drug pipeline, and this represents all new compounds that are in development from all different organizations, academic institutions, or other pharmaceutical companies that have been moving through early stage and clinical development. Next slide. So just to tell you a little bit about pediatric TB formulations, on the left here, you'll see what traditionally was the method of treating childhood TB, which was with adult pills that were broken, crushed into powders, mixed into any kind of food to give to a child of, all ages. And in doing that, it was very difficult to find the proper dose for a child at their weight range. And the pills taste terrible. We already know that these pills have a lot of side effects. In addition to the heavy side effects, trying to put these pills into a yogurt or anything else is just not great for kids. So we worked with WHO and Unitaid and some other groups to develop a formulation that would be properly weight banded. So it was tested in children, and a formulation that was, a single pill that's dispersible in a fluid that can be in water or another fluid that is accessible to patients. And it's the proper dose for every weight band of child. This is now available everywhere in the world where we have a lot of TB. I'm going to go to the next slide and I'll tell you a little bit more about accessibility here. But, over 2 million children have been treated with or have access to this treatment. And that's the number of products that have been procured around the world for treating TB. It's in over 123 countries. It's estimated that it's being used to treat about 75% of the global childhood TB burden of those cases that are identified. And something I want you all to be aware of is that this is not available in the United States or in Europe or in Canada or in high-income countries. And one of the reasons why that's true is because the regulatory authorities require different formulations to be approved in a new geography. In the United States, physicians also can't procure products from the Global Drug Facility at low cost. So we're in a situation where we have kids with TB in the United States that are still being treated with those arcane methods of crushing pills. And it's really devastating. So we're partnering with TB physicians across the US and working with some other advocacy partners to find a way to introduce these products in the United States. This is a point for advocacy, and it is something that I think we definitely want our lawmakers to be aware of, that this is something that needs to change. Next slide. The other products that we've already brought to market is, as I said, BPAL, which is being used to treat drug-resistant TB. On the left, you'll see the number of pills that are in a regular BPAL regimen. On the right is a standard number of pills that used to be taken for drug-resistant TB. Very long treatment times, hundreds, tens of thousands of pills. Now we have a 6-month all oral treatment to treat drug-resistant TB. Next slide. And this is also available everywhere in the world. 226,000 patients can be treated with these drugs. The amount of drugs that have been procured, which is one of the ways we measure how much is being uptaken in countries, is through procurement numbers. And we know that 226,000 courses of this treatment have been procured in over 110 countries. And again, the estimate is that we're able to treat about 80% of the global burden of drug-resistant TB, and this is just within 3 years post WHO recommendations. What I want to just go back to is something that Lizzie said about point-of-care diagnostics and the cases that we're identifying. When we talk about how much of the global TB burden is being treated, that's of those cases that are identified, and we still are not finding all the cases. That should be remembered. Yes, moving on. I just want to introduce a new product, which is sorfaquilin. This is another compound that's in development. It's just finished phase 2 trials, and we're making plans to move it into a phase 3 trial. Sorfaquilin is a diaroquinolone, which is the same drug class as bedaquiline. And we know that bedaquiline is a cornerstone drug of all important DRTB treatments right now. And we also have seen that because bedaquiline has been used for over 10 years in the global population, there is some emergence of resistance to that drug. That's normal. The TB bacteria is incredibly good at mutating and becoming resistant to drugs. And so I think when we think about developing new drugs and making sure that we're always prepared for what's next with TB, one of the things we have to be prepared for is making sure we have a healthy size pipeline, because new generations of drug treatments are going to need to be developed to make sure that we're catching all of the TB before it becomes resistant to medication. So sorfaquilin, or TBAJ876, sorfaquilin is the new drug name that we're all trying to get used to and figure out how to pronounce, is basically a next generation diaroquinolone that could be used in place of bedaquiline in a treatment. We've seen from earlier studies that it is more potent. It may have fewer side effects like QT prolongation. It is potentially effective against strains of TB that are resistant to bedaquiline already. And so it's quite a good candidate to move forward for a next generation treatment. Next slide, please. We just finished conducting the NC09 trial, which was a phase 2 trial where we were doing many things in one trial, but we were trying to find an optimal dose for sorfaquilin in pairing with pritomanid and linezolid for treatment in this trial of drug-sensitive TB. And we were comparing that against BPAL in a drug-sensitive population for 6 months, and also HRZE, which is the standard of care for drug-sensitive TB. And we've shared some of the interim results most recently at the Union Conference in Copenhagen last year. We do know that there is a dose that seems to be optimal, which is 100 milligrams. And in combination with pritomanid and linezolid, that did outperform BPAL and HRZE in a 6-month, 4 to 6 months treatment in drug-sensitive patients. No specific drug-related safety issues were noted, and overall the side effects seem to be less. I'm not going to get too much into the details of the technical outcomes here because I'm not one of the clinical colleagues at TB Alliance. And if you do have questions about that, please let me know and I'll get you answers to that. But next slide. This is just a visual of where we've been with TB treatment, where we're hoping to go. All the way on the left is our historical treatments of, up to 18 months to 2 years of treatment with tens of thousands of pills, sometimes injections to treat all forms of TB and resistant forms of TB. With the introduction of BPAL, we've been able to reduce the length of treatment from those 18 months to, and there was a previous 9-month treatment as well, but now we can treat most cases of drug-resistant TB in 6 months with just oral pills. What we're looking at with SPAL or sofequilin with pratominid and linezolid is potentially 4 months treatment with fewer pills. And the next stage of clinical trials is to test this in a drug-resistant population and drug resistance of all severities to compare that to BPAL, which is the current standard treatment for most forms of drug-resistant TB, and to do that comparison. To reduce treatment length more to 4 months would be great, but to really revolutionize TB treatment and get past this, burden that we are seeing year after year globally and the number of deaths that we're seeing is to really change it so that it's much easier for people to take. And what we've seen with the emergence of the use of long-acting injectables in HIV treatment, where you can use a liquid formulation of a drug and use an injection to have a sustained level of that drug in your body for a long period of time, opens the door for looking at that possibility in TB treatment as well. So TB Alliance is actually doing in parallel to the drug developments and moving oral drugs through clinical trials. We're looking at formulations of these same products in long-acting injectable formulations so that they could potentially be combined. So if you go to the next slide, I'll give you a sense of where, we put this slide up and it's very exciting to say we see a future now of TB treatment in potentially 1 day for latent TB infection or 1 month for all active cases of TB. And we wouldn't say that unless we thought the science was not really there. And that's what's exciting right now about TB research, is that there has been so much progress and we are seeing a lot more products in development that we are starting to see the potential to get to this place in 5 to 10 years, we hope. So what this would look like is, as I said, 1 month of treatment. That would be a combination of oral treatment for about a month and a long-acting injectable treatment that would stay in the body for several months to treat active forms of TB, and potentially just a 1-day treatment for latent forms of TB. So that is where TB Alliance is headed. Next slide. I can't remember what I have next, but it might be the end. That's the end of this presentation. This is all of our funders. We do receive US government funding. As Lizzie said, it's been very difficult over the last year to have that sustained, and we're very grateful for a diverse set of donors to make sure that this work can continue without interruption. Thanks. And with that, I'll introduce Kelly Kern.
Thanks. That was fantastic, Stephanie. My name is Kelly Curran, and my presentation, I think, is going to expand a bit and give a few examples of some of the work that our colleagues at the TB Alliance are doing. I work at the Johns Hopkins School of Medicine in the Center for TB Research, where I am the project director for a project that's called SMART for TB, which stands for Supporting, Mobilizing, and Accelerating Research for TB Elimination. And this is a US government-funded award. It was a USAID-funded award that's now managed by colleagues at the State Department. And it brings together a consortium of researchers, educators, and public health experts from around the world. And we're currently working on a 5-year timeline that ends in July of 2027. Again, like many US government-funded projects, we've gone through a few changes over the last year. And right now, we can go to the next slide. We are focused on implementing 5 clinical trials. 2 are in the area of diagnostics. I think Lizzie did a really nice job of kind of laying out a lot of the challenges that we have with diagnosis. It's actually where in some ways, the biggest challenges are in the tuberculosis treatment cascade is just making sure that people get diagnosed correctly and promptly to begin with. This is, as Lizzie mentioned, it's really especially a challenge for children who oftentimes it's difficult to get them to produce sputum. And so our work in diagnostics is looking at kind of alternatives to current sputum-based testing. And then we're also implementing 3 clinical trials in the area of therapeutics. And we'll go into those. One is a pediatric trial. One is focused on drug-resistant treatment for adults. And then the third trial is actually a prevention trial, but we're kind of lumping it under the area of therapeutics that is looking at a new type of TB preventive treatment for people with latent TB or who are at risk of developing TB to prevent them from developing active TB. Next slide. So ADAPT is the name of the trial that is looking at new diagnostics in adults. It's currently working in 3 countries, Nigeria, the Philippines, and Zambia. And this trial has been it's led by colleagues of ours at UCSF and UC Irvine, working with a number of partners. And they have been doing, in the first 2 years of Smart for TB, really focused on looking at tongue swabs as a new way of collecting samples to test for HIV and looking at a number of different platforms that are testing for tuberculosis using samples collected through tongue swabs, which of course are a lot less invasive. And as Lizzie mentioned, not everyone can produce sputum. But the tongue swab is essentially, a swab is maybe a little bit of a delicate way of putting it. It's kind of more of a tongue scraping, to be quite honest, but it's something that you can obtain a sample pretty much from anyone, whether they're able to cough up sputum or not. So next slide. Adopt for Kids is the sibling to Adopt for Adults. And again, this is a study that is really focused on trying to find new ways to detect tuberculosis in children. TB is actually a top 10 cause of death in children around the world. And as was previously mentioned, it's not easy to diagnose tuberculosis in children, and oftentimes this means they're getting diagnosed late, if at all, and sometimes, even passing away before they have had an effective diagnosis. This trial is currently working in Mozambique and Uganda, and the focus is, again, looking at new point of care or near point of care ways of identifying tuberculosis in children, including looking at tongue swabs, stool and urine-based sample collection, as well as some sort of technology-based tools like computer-assisted software for analyzing chest x-rays. And as well as an AI cough app. I think Lizzie mentioned this as one of the innovations in diagnostics, so that actually listens to the way a child is coughing and hopefully will be able to differentiate between a tuberculosis type of cough and a cough that's caused by a different kind of respiratory infection. So this is kind of an exciting line of work. And this study is actually linked closely to our pediatric treatment study that I will talk about. I believe that's the next slide, which is SMILE. SMILE is the shortened regimen for drug-susceptible TB in children, or SMILE TB. This is a study that is focused on treating children with drug-susceptible TB, so not resistant TB, either kids with pulmonary or lymph node TB, and trying to assess whether half the length of treatment, so shortening treatment to 2 months from the current standard of care, which is either 4 or 6 months, depending on the country, to see whether the new short course is as effective in curing TB in children as the current standard of care, which is longer. I think Stephanie did a great job of discussing the challenges with pediatric treatment, the challenges of not always having dispersible medications, the taste. And so I think we can all agree that, shortening the duration of treatment to 2 months will probably increase the percentage of children who are able to complete treatment successfully. So this study is currently being implemented in India, Indonesia, Mozambique, Uganda, Zambia, and Zimbabwe. We are actively enrolling and looking forward to being able to answer this question once we fully enroll and are able to follow kids out long enough to be able to determine whether the short course is as effective as the current standard of care. Next. So PRISM is a study that is focused on the treatment of drug-resistant TB in adults. Again, Stephanie talked a lot about BPAL-M, which is the current backbone treatment for people with drug-resistant TB. And while there has been a lot of progress in shortening the duration of treatment, I think Stephanie did a really wonderful job of kind of laying out the goal here is like, how low can we go in terms of duration of treatment, given that it is still burdensome for folks. So PRISM is a study that is actually taking a stratified medicine approach to people with drug-resistant TB. And so there's an algorithm that is assigning people points for their sort of risk of needing longer treatment versus shorter treatment, if that makes sense. So we know, for example, that people living with HIV as well as TB might benefit from a longer treatment, whereas people who are HIV negative may be able to respond to their TB treatment in a shorter period of time. Likewise, the just bacterial load is oftentimes a correlate of whether people are able to respond to TB treatment in a shorter or longer period of time. Basically, the problem that PRISM is trying to kind of solve for is that right now, many clinicians who treat people with drug-resistant TB estimate that some people are getting overtreated. So, they're getting treated for 6 to 9 months when they probably would be cured in a shorter period of time. But then there's still a subset of people who are probably being undertreated for whatever reason, their current health status or their age or something about them, and their health means that they might need a longer goal. So this is again, PRISM is trying to set out to establish whether with a kind of algorithm and a point system, we can better assess who can benefit from a shorter treatment or a longer treatment. And this study is currently enrolling in Mongolia with sites about to come on board in Peru and Uganda. And then my final slide is BREACH. BREACH TB stands for Bedaquiline Rollout Evidence in Contacts in People Living with HIV to Prevent TB. So again, this is a TB preventive treatment trial. So for people who have latent TB or TB infection, and they're at risk of developing active TB, but they don't currently have active TB. As I think, again, I'm happy to talk about this because Stephanie did such a nice job of talking about how the goal is, again, to shorten not just treatment duration, but also the duration of TB preventive treatment. And so this will be comparing 1 month or 28 days of bedaquiline, a drug that's been mentioned already quite a lot this evening, to the current standard of care for TB preventive treatment. And the goal here is to develop a sort of pan prevention treatment. Stephanie was talking about a pan treatment regimen. Right now, if someone is exposed to drug-resistant TB, for example, they are usually given a 6 months course of TB preventive treatment. If they are exposed to drug-susceptible TB, they are given usually 12 weeks of treatment through something called 3HP. So again, this is a study that is trying to see if we can shorten the duration of TB preventive treatment to 28 days of oral bedaquiline. And what's, I think, really relevant about this study as well is, as Stephanie mentioned, there's been a lot of effort to develop long-acting formulations of these medications. There is a long-acting formulation of bedaquiline that is in the pipeline. And so I think the BREACH study will also provide some evidence about whether whether bedaquiline can be used as a TB preventive treatment, because the 28 days of oral bedaquiline is very aligned with the single shot long acting bedaquiline that is under development. Breach is not yet enrolling, but will be enrolling participants in Mongolia, Peru, Tanzania, and Uganda. And that is it for me. Just want to thank everybody for your interest. I know it's a late night, but we'll go on to the next colleague now.
Thank you, Kelly, and really happy to be here. My name is Anna Maria Harkins. I'm with IAVI, another product development partnership focused on vaccines and also antibodies. We develop a host of vaccines, including TB, but also HIV and a portfolio of emerging infectious diseases. Like many product development partnerships, we've been around for over 2 decades, and this partnership model really depends on the community, on research partners, and on the advocacy community that's on this call. So we really appreciate working in concert with all of you to advance this goal. You could go to the next slide. So I just will briefly address the fact that there actually is a vaccine. But I think, as most of the people on this call know, it's a super important vaccine for infants and young people, but it is less effective for adolescents and adults, and it really doesn't have very much effectiveness once you enter adolescence. So the real work ahead, and again, as part of WHO's roadmap to end TB, is to have multiple effective vaccines that work over a host of different populations and that are accessible to people who need them in low and middle income countries, as well as the global north, and that it's part of a comprehensive package of care. You know, it's 3 legs of a stool here. We have drugs, we have diagnostics, and vaccines, and all of them are really equally important for us to be able to tackle this as a whole. You can go to the next slide. I'm going to zoom out a little bit and talk more about what is going to be really important to talk to legislators about and Congress, and that is that this is a massive public health issue and it has socioeconomic impacts for everyone. There are studies, and I think when you get copies of the slide, you can click on the hyperlinks to look at the source, but the estimated benefits over 25 years in a cross-section of countries really shows that even if you have a vaccine that has 50% effectiveness, in adolescent and adult populations, you're talking about millions of disease lives averted, deaths averted, and also treatment averted. And that really translates into dollars in a very, very significant way. When you think about the amount of money devoted to TB treatment and diagnosis, not just in low and middle income countries, but in the United States, some states budget for TB. If you have 1 to 2 folks who have multi-drug resistant TB, that blows the entire budget for their allocation. Next slide. Again, looking at TB vaccine research and development, as TB writ large, TB vaccines are no exception in the sense that they are chronically underfunded. And not only are they chronically underfunded, when you look at this chart, you can see that there isn't really a very healthy diversity of donors who are contributing to this. And I have to say, in the past 12 months, this pie chart, I mean, this is from 2023, so I actually am afraid to recalculate this pie chart right now. But what you see is philanthropy is really outspending bilateral governments in the development of TB vaccines. You see, of course, the Gates Foundation and Open Philanthropy that are doing really the lion's share of a lot of funding for vaccines. You do have the NIH, which does a lot of the early stage research, and then you have these bilateral governments. But what we really want to see is, number one, a much, much bigger pie, but you also want to see more slices in that pie. So this is really, I think, an important advocacy message that we need to give, is that the funding matrix is very, very fragile, and it really needs to be bolstered, especially because, as we've been hearing over the past 45 minutes, the science is incredibly exciting, but the funding is just not keeping up with the science. Next slide. So this is the global TB vaccine pipeline. I won't go into it, but maybe just zoom out to say that what you really want to see are boxes across this, but many boxes. You want to see plenty of preclinical candidates coming into phase 1. You want to see, a healthy, diverse group of potential trials going into phase 2. And then you want to see lots of phase 3 trials. But what you actually see is not quite as healthy as it really needs to be. There are some good aspects to this. There are a diversity of vaccine platforms, and what I mean by that is, if you look, there's a live attenuated, there's viral vectors, there's protein, there's mRNA vaccines, and you want that sort of diversity of different approaches to developing a vaccine. But what you really see, especially on the right side of this slide, are 2 trials that are efficacy trials. We really should be seeing multiple efficacy trials. Just think back to the COVID-19 pandemic. How many trials were happening for COVID-19 simultaneously at risk, right? That's what unlimited resources looks like. And I think, we need to be ambitious in the way that we characterize the need for funding. And we need this pipeline to look significantly more robust than it does right now. You can go to the next slide. Oh, it's a fade. You can go to the next slide. So let me touch on the 2 efficacy trials that are happening now, and I'll start with MTB-VAC. This is a live attenuated vaccine in development. IAVI is the sponsor. It is owned by Biofabri, which is a pharmaceutical company based in Spain that's part of a larger pharmaceutical group in Europe. And the objective, as I said, is to evaluate the efficacy of MTB-VAC in preventing pulmonary TB disease. It has a full range of antigenic targets on the original pathogen. Again, it's a phase 2b trial, not a phase 3 trial, but we've really tried to pack as much as we possibly can into this trial to answer as many questions as we can. The goal is to enroll 4,300 adolescents and adults across 5 sites in South Africa, Kenya, and Tanzania. We enrolled the first patient in February of 2025, and we really look forward to being able to continue that enrollment. Much like actually M72, which I'll speak to next, a lot of epidemiology work was done in advance of this trial to be able to carefully calibrate enrollment sites and catchment areas to make sure that we could enroll the people that we really needed to enroll. Next slide. Okay, so one more slide on MTB-VAC. If you want to just go to the previous slide, thanks. The access profile, I just want to speak about this because it's something that's really been very heartening, and I won't give my own spoiler alert to what I feel most heartened about and optimistic about. But it's a very interesting vaccine. It has broad antigen presentation, a non-adjuvanted formulation. There's an ease of administration that we think will go a long way for access, and we have multiple global manufacturing partners already lined up, which is really important. We have BioFabry, which will be in predominantly high-income countries, but then a Brazilian manufacturer, and then Bharat for South and Southeast Asia and Africa, which is really important to have those commercial partners lined up. And of course, as always, we have an absolute commitment to affordability. So there's been a lot of work done on cost of goods, and the work will continue actually. And I'll say, just a plug, that this kind of work about acceptability and affordability is so important to do pre-licensure. It's really, I think, a best practice, and it's something that we've learned the hard way over time. Okay, now next slide. So M72 is the phase 3 trial that the Gates Medical Research Institute is spearheading. It's got funding from Wellcome Trust, the Gates Foundation, and also Open Philanthropy, now called Coefficient. And it's, again, an efficacy trial to test prevention of pulmonary TB disease. The phase 2b trial results were really, really encouraging. There was an estimated efficacy of 50%, and that's also what the target is for the phase 3 licensure trial. GSK licensed the antigen to this, and GMRI has control of it, but GMRI retained control of the adjuvant. This is enrolling 20,000 adolescents and adults, and they are, I think, well on their way to completing enrollment, which is absolutely extraordinary when you think about it. So it's really been something that has been a tremendous achievement, and I can't wait actually to see the interim results. Next slide. Just a word about, community engagement and the importance of nothing without us. So just many, many things have been in the works as part of various forums and consortiums, and there are just a few here. Again, these are all linked. Again, shout out to TAG for What Could Go Right publication. It's really great, and I encourage you to read it. But all of these boxes really just are about the central message of ensuring that communities are part and parcel of driving this bus, to make sure that ownership is guaranteed and that there's transparency and fairness in this whole process. I think that might be the next slide, is hopefully just thanking, of course, to all of the funders who make this work possible. Thanks.
And I think that might be me on the next slide. And Anna Maria, that was a really great segue into what I wanted to talk about in terms of community engagement. So I think a lot of the time, especially for advocates like myself, the way that we think about community engagement is more on the, I guess, kind of feedback end. But that is not strictly the case. And I think community advisory boards are a really important example of that. These are structures in which folks who have relevant experience with the topic at hand, whether it's from personal lived experience or professional experience, folks from civil society and community, are able to provide direct input on research everywhere from protocol development, which is sort of like the pre-research stage where things are really being written out and planned, all the way through to implementation. And just as some examples of community advisory boards that TAG is involved with, we have the Global TB CAB, we have the Community Research Advisors Group, which is a specific community advisory board for one research consortium. And then there are other CABs that are specific to certain geographical regions, like APCASO, AfroCAB, and ECAT. There are also CABs that are specific to certain subpopulations. So part of PregLAC's work is with people with lived experience of TB during pregnancy and lactation. There are so many different ways that community can be defined, and that folks can come together to really provide that important expertise, not just as a sort of box to check off, but really as an accountability measure to make sure that whatever is being researched is as responsive and as accessible and as needed to community as possible. And the next slide, I'll just close with this. So really want to bring it back to the fact that all of this goes back to human rights, and based on the UN recognized Declaration of Human Rights, there is a right to health, and within that right to health, there is a right to science, which means the right to enjoy the benefits of scientific progress and its applications. And when we see a lack of funding for research, when we see a lack of support for research, when we see a lack of accessibility to the products of research, those are all human rights violations. And I think it's important to center our conversation around that core fact that what we're talking about here can sound very abstract because a lot of it happens in labs, and a lot of it is talking about a mycobacterium and how it responds to different biological agents and or anti-biological agents, right? But what this is really about is human beings and human rights. And when folks like you are engaged in this advocacy, I want that to be kind of the driving force behind having TB R&D involved in that advocacy. So that's all.
I did the thing. I forgot that I was on mute. It happens. So that was amazing. And we have like literally dozens of questions that have come in through the chat. And I know we're not going to have time for all of that, but I'm hoping that potentially there's a bunch of these that maybe when we send out this link for people who aren't able to make it, we can include some written responses to some of these questions as well. So I am just going to get started because some of these are really great questions. And I am going to start with one of the fun ones that is very selfishly my question for Kelly, but there was a lot of chatter about this in the live stream chat. Who comes up with these acronyms? Because they are amazing. And like, do you start with a word and fill yourself back into the acronym, or like, how does that happen?
Oh, well, this is also this is like one of the few times that I would say that scientists get to be really creative and literary is when they're coming up with names for their studies. So oftentimes, there's kind of a brainstorming amongst investigators, but I think, Lizzie and Anna Marie and others have talked really eloquently about the importance of including community voices in the development of trials from the very beginning with community advisory boards and others. So I'm glad everybody likes the names. I think it's important, to have, especially if a trial is going to become really seminal and important and really game changing, which is what, of course we want. Like, for example, Smile TB, the pediatric trial, like it will put a smile on my face if we can shorten pediatric treatment to 2 months, And so I think having a memorable name is also helpful. So when we're going to talk about what we hope to be game-changing trials. But thanks for that question.
Yeah, no, that is where it's like that is hard to do. It's hard to come up with that kind of thing. So in addition to the really difficult science and that's going on, we want to make sure we acknowledge that part. The rest of the questions are less fun, and I do apologize for that. But the first one is one that it's two parts, but I think it's a common theme in here, which is, do we struggle like at companies to start on new trials for drugs and tests and vaccines because it's not profitable? Like, is that a capitalism problem in there? And on a similar note, are we seeing a slowdown in new research just from people not knowing whether or not the funding they have today is still going to be there tomorrow? Who wants that one?
I mean, I'm happy to start off and then others can chip in. So I'm actually pretty new to TB. I have spent most of my career working in HIV. Almost 25 years working in HIV before I moved over to TB. And obviously the two diseases and the two pathogens are very linked, but I think the response has been really different. And something that I have seen in my years is that, HIV has always attracted a lot more private sector investment, pharmaceutical investment, because there is a market for treatment and for prevention in high-income countries. And while TB does affect some people in high-income countries, it mostly affects people in low and middle-income countries. And so I do think, and again, here I'm really speaking as a person, and these are my own thoughts. But TB treatment, it's now the leading infectious disease killer for a single pathogen. When I started working on HIV was far and away the leading infectious disease killer. And that's changed. And that has changed because there have been incredible advances in diagnostics, in therapeutics, and in preventive treatment like long-acting lenacapavir and others. And so but part of those, those were investments that were made from both public monies and donor philanthropy monies, but also investments that pharmaceutical companies made because they thought they could recoup some of their investment. And I think in TB, there's less of a profit motive, and I think that's why organizations like TB Alliance, which is a not-for-profit, are so important. Because TB treatment is a public good, that we need to have as a world. Everyone has a right to health, and whether or not a profit can be made on a diagnostic or treatment. That's my own personal opinion as Kelly Curran, not speaking for any donor or employer. I don't know if others have something to add.
Sure. I can add just from, developing drugs and moving into clinical trials. I think, we're in the not-for-profit business at TB Alliance and IAVI and other PDPs, and there's a reason for that. I mean, these products don't have a market. They should be available to everybody. But that doesn't mean that the research is not incredibly expensive to conduct. And when you're developing new products that are not already approved in the market, that means you need to do clinical trials at a standard that regulatory authorities will review the data from those. So registration trials can cost a lot of money, and that's not dissimilar from what it might cost a pharmaceutical company to do them, but we're a not-for-profit doing them. Another aspect of doing clinical trials in this kind of environment that's really important is that they do need to be international trials. They need to have multiple sites, multiple geographies, enrolling individuals from many different regions, make sure that we have the best chance of testing this in a broad population that's affected by TB. So doing those kinds of trials are also very expensive. I think one challenge, I think Anna Maria said this perfectly when talking about what a healthy pipeline should look like. And we know what it could look like if there were unlimited resources. And we've actually all lived through that kind of recently. And that is kind of boggles the mind that we are still struggling with so many other things, and we know the answer. So it's not a limitation of the science. It's not a limitation or resistance because there's not going to be profit there. It's really limitation of resources to do the work. But the PDP can only go so far. Even though we do participate in making products accessible to the public and make sure countries can take them up, it does also cost money to make them. So it is hard in this environment to find manufacturing companies that are willing to commit at risk to developing products once they become approved by the US FDA or the EMA or other regulatory authorities, and kind of see how that uptake goes. So it still continues to be challenging, and we would like to do a lot of this more quickly if the funding was there.
There any other builds on that one? Because I have a related question that is in here, two related questions actually. One of which is, and there's a solid chance we don't know the answer to this yet, but the leadership shakeups at the FDA, have they changed how companies are navigating the approvals process, and what are the challenges coming from that? And we can also just skip this one if we need to, because we just don't know yet.
Well, actually, I think we do.
Oh,
I mean,
I was hoping that maybe there might be a bright side that we haven't thought of yet. Unlikely.
if you think of it, please call me.
Yeah.
I mean, certainly for vaccines and vaccine makers, I mean, I can't wait for the World Vaccine Conference this year. But what you want from a regulator is clarity, right? Either clarity in your favor, but even if it's not in your favor, at least you are clear about it. But what's happening now is total and absolute chaos. So you will find that product developers seek it from the European Medicines Agency, which means that you can't get it approved in the United States and you won't have access to it, or they just won't develop it at all, or they'll just shelf it. So, I think we actually are watching this in real time.
Yeah. So unfortunately, the answer is it just sucks. Okay. So one of the other things that's come up, because we have a lot of very enthusiastic TB advocates, very few of whom are scientists. So we were hoping we could get a little bit of information on like the various phases of trial, and we might need the super abridged version for now. But when we talk about phase 1 versus phase 2 versus phase 3, what does that mean in terms of what's happening, how much that costs, and how long that typically takes?
Who wants to tackle this one?
Like, that's a lot of question in one.
So I have sort of one piece of input on this, but I think other folks have much more sort of in-depth insights. One thing I'll say is that more or less, as you move through the pipeline from preclinical to clinical work, conducting each individual trial starts costing more and more. But you are able to narrow down which trials you're conducting. So it's not that there's need for more funding later on, because in the preclinical work, you're not exactly throwing spaghetti at the wall, but like you are trying to find, I'll just like keep speaking in metaphors. You're trying to find a needle in a haystack, really. And so you are spending more money on something that each individual time is much less expensive, but you're having to do it a greater number of times, if that makes sense. And then as you move through the pipeline, you're conducting fewer of these individual things, but each time you do it, it gets more and more expensive because you're enrolling more and more people, and you're also, when you move from preclinical to clinical, you're actually involving people rather than a petri dish. Yeah, but I'm sure other folks have other more relevant details to include on that.
I think that was a great start, Lizzie. As I said, I'm not one of the clinical people, so Kelly, please jump in to support me in this. But just to go through the basics of what happens in each stage of clinical trials, I think it is important to understand at what point are clinical trials enrolling people that actually are sick, and what we're trying to find out in those trials. And so, if you think about all the preclinical work, like Lizzie said, is either happening in a lab or it's happening in animal studies, and you're not testing something in humans until a phase 1 clinical trial. And, if typically before you want to test a new medicine in someone who's actually sick with something, you want to make sure that it's acceptable to the body. And so you may test it in healthy humans. I lost my train of thought there. Oh, right. So when testing in healthy humans, you're basically seeing like, well, how does the body metabolize this medicine? What's going to happen if I take this in my mouth, and is it going to come out from here or there? And how long is it going to be in my body? And what is it going to make me feel like? And then when you're testing in people that actually have disease, you have to, that's testing for safety, and also kind of figuring out what it does when it's in your system. But what's really important is that new products are safe and effective. So when you're talking about testing things in people with disease, they are being tested for efficacy and safety, tolerability. So really looking at every aspect of what's happening with the drug product when it's going through the system, and how that participant is reacting to it on a very regular basis. So, and in the process of having people with disease in a clinical trial, you're also caring for those individuals and their disease typically. So, we can't just test a new treatment on someone with TB. We're caring for that person with TB and making sure that they're cured in the end. So that's a commitment. It's a long process and involves a lot. I'm going to go to Kelly. I just wanted to say one thing, if you'll come back, about how we're trying to make these processes more efficient so we don't have to do so many of them. Yeah.
No, I think it's such a good question, and I really appreciate the point that was made that even before we get to a phase 1 clinical trial, which is the first time that a new product or drug is being tested in humans, there's a huge amount of work that's done in the laboratory and sometimes with mouse models. Like some of my colleagues in the Center for TB Research are working mostly with animal models to make sure that drugs are safe in other mammals before they get tested in humans. But in general, a phase 1 trial is, again, the kind of early safety, and as Stephanie mentioned, it's usually conducted in healthy volunteers, healthy participants, so who may not have the disease. And then this phase 2 is and that's a pretty small number usually of people. The phase 2 is a slightly larger number that continues to look at safety, but phase 2 also works really hard to try to establish dosing, especially if it's a drug trial, so what would be the appropriate dose, and starts to look for initial indicators of whether that let's say it's a new drug, is actually effective. And then the phase 3 trial is the efficacy trial, and that's usually involves a much larger number. Anna Maria was talking about the vaccine trials are enrolling, thousands and maybe tens of thousands of participants, because in a prevention trial, like a vaccine trial, you actually need to enroll enough people to capture enough people who might develop TB along the way, to test whether there's a difference between the group that got the active vaccine and the group that got the placebo vaccine, if that makes sense. So in general, phase 3 trials are quite expensive to run because they involve, thousands usually of participants. And as Stephanie mentioned, all those participants are getting a high level of care, a standard of care. I can tell you, for example, the BREACH trial that we're doing on TB preventive treatment using bedaquiline, all the participants need to have an EKG to make sure that, the medication is safe for their heart. That's something that often isn't a standard of care in a normal like primary care setting in most African countries, so they're getting but we do need to know, obviously if this medication that we're giving to healthy people, because a prevention trial, by definition, you're giving a medication to somebody to prevent a disease, you need to make sure that it's going to be very safe for them. So I hope that but I mean, there's efforts that are being made. We also, I will say, in Smart for TB, we've made a few changes to the structure of our trials. So both PRISM and BREACH actually right now are kind of structured as like phase 2 and phase 3 trials together, so we're going to basically make sure that we have a strong signal that things are looking promising before we go to a full efficacy trial. I think there are, a lot of biostatisticians that we work with are really trying to think creatively in this current environment where funding is really constrained. Like, how can we use resources as effectively as possible to answer these scientific questions as quickly as possible, but in a way that conserves funding, recognizing that big, huge phase 3 trials are really expensive to run. I don't know, Stephanie or Anna Maria, do you have anything to add there?
Yeah, I think you addressed what I was going to say is like changing clinical trial designs to make sure we can do as much as possible in one clinical trial so that we don't have to do them sequentially and can do them a little bit more in parallel to save time, but it can also really save resources.
Yeah. Thank you all for that, because I think one of the things we hear a lot is these trials are big and expensive, and it's hard to quite comprehend, like, but why? Is it just the cost of the drug or the treatment itself? But no, it's there's so much more to it in that administration, and it takes so much work. And so it's nice to understand all of that a little bit more too, and give some recognition to the people actually doing it.
Mm-hmm. I mean, I can just say, since we're running, clinical trials right now, the cost of the medications in most cases is not the big driver of the cost of the drug. I mean, it depends on, I think, for some really new agents that might be different, but for example, the studies that we're doing are all with existing drugs that are already approved. Obviously, BREACH is looking at a new indication, prevention rather than treatment, but the costs are, all of the sites that are doing the trials, they need to have doctors, nurses, pharmacists, community health workers that can go. What if somebody doesn't come back for their next visit? They need to have, that community health worker needs to have a cell phone with airtime to go to try to follow up and find out, did that person not come back because they're sick? Did they move? Like, what's going on? So there's there's a lot that goes into, and I can say this as I worked on PEPFAR, HIV programs for years, where we were kind of serving huge numbers of people in a programmatic setting, in a kind of high throughput model. And clinical trials is like the opposite. It's a high intensity, small number of people, but it needs to be a very controlled and high quality setting so that you can really answer the question, but there's a lot of good thinking that's being done about this, and a lot of people who are really also trying to make sure, that while clinical trials are very controlled settings, we also want to conduct them in as close to the real world as possible, so that when we're, assuming that whatever we're studying reports out positively and hopefully will get approved in guidelines, we want to make sure that it's as easy as possible to translate that finding into real world programs, if that makes sense. So, that starts with trying to test things that are feasible, and acceptable to participants.
Yeah. Well, that is really great insight. I was thinking like the cost of keeping track of the paperwork for 20,000 people alone sounds like a nightmare, let alone all of the other pieces. But on the subject, like while we're thinking clinical trials and everything coming forward, I think this is a good spot. We have more questions that we'll find ways to get people answers on, but we're well over time. So I just want to do a quick round robin, because we here at TB Fighters are very, very big fans of hope. We think that's kind of the only way to get out of this situation. So I just want to kind of go around and find out what is keeping people optimistic in the world of TB right now. And Stephanie, you're at the top of my screen, so I'm just going to start with you.
Oh, yay. I get to go first. Well, I said this during my presentation too. I'm incredibly excited and hopeful about the science that we have now. And so I think that there's so much potential for what we'll see. But this is a group of advocates, and what I think that I'm just so I think what's very hopeful is the number of people that are showing up and advocating for TB. And this is those of us who've been working in TB for a couple of decades did not see this at the beginning, and it's just a whole new world. So the struggle is there every day, but it's easier to do it together, and we really appreciate it. So that gives me hope.
Anna Maria, you're off mute. Do you want to go next?
Yeah, I mean, I feel like we're going to sound like a broken record, but I too am super excited about the science. I'm excited that we've advanced on all fronts to really have products that are either in the market or looking we're within striking distance, relatively speaking. And also, yes, I think a lot of that is attributed to people's just determination and tenacity to not give up, to keep at it, and to be persistent, and to, you know, advocate in a very forceful way. And guys, it's working. Despite everything, it's working.
It's hard to remember, but we've got to keep saying it. Kelly, why don't you go next?
Yeah, I mean, I really share so much of Stephanie's enthusiasm for what the TB fighters are doing. It's so amazing, and I can't wait to meet so many of you at Hill Day in early March. Um, Again, I mentioned that, I'm coming into TB after working on HIV for 2.5 decades, and seeing that when science, advocacy, and political will line up, like those three legs of the stool are line up, you can change the course of an epidemic or a pandemic. And I had the great privilege of living through HIV changing from an automatic death sentence that was killing between 2 and 3 million people a year and orphaning tens of millions of children to a manageable chronic disease that kills, only a little more than 500,000 people a year, still far too many, but the mortality has been decreased by 2/3 or even 3/4. And the same can be done for TB. But we needed better tools, and HIV had great diagnostics and we had great treatments, basically. And once we had that, everything kind of it was much easier to achieve those sort of gains. And everything that happened in HIV happened because of advocates and activists and people who fought and who argued for funding and who argued for better science and who argued for more community involvement in the science and in the programs. And I am feeling like TB is really at the cusp of changing the narrative, and in a few years I don't believe that TB will still be the leading infectious disease killer, and that'll be thanks to a lot of the efforts of people who are coming to Hill Day. So thank you.
Yeah, and we also have so many advocates in our network that aren't able to come but are still doing all of the work on their end
Yes.
too. Like we're in it and very convinced
And
that
sending
it will
letters,
be
I will
TB
say,
wise.
yeah, if you aren't able to come, I know the letters are really meaningful to members of Congress. And yeah, you still have a voice, even if you aren't physically able to come to DC.
All right, Lizzie, we're going to end with you, and then I've got a couple of quick announcements.
Yeah, I wholeheartedly agree with everything that other folks on the call have said. I mean, working with TB fighters has been really a game changer in advocacy. I mean, you all are hope incarnate. And I'm not speaking in hyperbole. It's truly amazing to see. And I think another thing that's really giving me hope is just the results of all of this. It's hard to see when there are so many attacks every day on what we're doing and the people we care about, but people are living longer. than they ever have, and people have more options than they've ever had. And um, I think one of the best parts is that people with TB are demanding more than they ever have and expecting more than they ever have. And that to me is really the biggest thing. So yeah.
There are a lot of reasons to feel good in the long run. So that is the note we always like to end on. I have a couple of quick announcements, actually not as many as I usually have to get through, which is exciting for me personally. Our book club is just started reading Stigmatized: A Mongolian Girl's Journey from Stigma to Illness to Empowerment by Handa Ank Amagalan. Ooh, I did not do that right, and I'm very, very sorry. So we started that book on February 1st, but we're running through the end of March. So if you haven't started it yet, you can still catch up. Details on how to get that are in the description of the YouTube video. I'm really excited about this one. It seems like a great book so far. I haven't actually started it yet, but I did read the description of it, and I am very excited on this one. If you didn't manage to catch our TB 101 training that we had on the 9th, that is still available for people to watch, and we'll send that out again. That is a really great one with our friends at NTCA. Dr. Kelly gives a great presentation on it. It is really wonderful. I also just want to flag our friends at We Are TB. They run a regular and anonymous support group for TB survivors, and we just want to, point everybody in their direction. That because they are one of the best community resources, I think, for us here. They do that in both English and Spanish. So the link for them is also in the description on the video. And if you are coming to Hill Day, you will get to meet a bunch of their lovely members, because that really helps us keep focused on the incredible people in this community. And I think that is everything for me tonight. So with that, as they say in our hometown, don't forget to be awesome.